Okay, how about we get started then? All right, I'll kick us off. So, welcome everyone to this tutorial for the ICB Africa. And apologies, we could not. We had some technical issues last time trying to connect, but we managed to sort out those technical issues and we rescheduled it to the tutorial for today. And I'm aware that it's holiday in some countries, so um, uh, I'm very glad you were still able to find some time to attend. But um, uh, just to let you know that this session will be recorded for the benefit of others, so that they can, and yourself, so that you can review the tutorial at a later time. So just to introduce the uh, myself and my other co-presenters here. So we're Dennis, Uray, and Evelyn. We're based in Singapore, and we represent a few institutes, including A-STAR in Singapore. It's a government research institute, the National University of Singapore, and Imperial College London. And today we'll share, give a quick tutorial on a topic of research that we work on, which is mainly predicting drug response. And this will hopefully give you an introduction to this kind of machine learning problem and an approach to tackling this problem using graph neural networks. Okay. Just some ground rules for this tutorial. Feel free to speak up anytime if you have any questions. So I think you should be able to unmute your microphone if you have a question, or you could just type into the chat box your question. One of us will be speaking, but the others can monitor the chat if you have any questions. Okay. And also, given that this is done over the net and we have latency issues sometimes, please let us know as well if if our voice cuts off or the screen sharing isn't clear. We'll slow down and try to repeat some things if anything isn't clear. Okay. So this is an interactive tutorial, so you can follow along as we go through the material and the examples. And also, it's encouraged that you go through the material later on at your own time, because we will go at a certain pace, and you may not be able to follow everything. But we'll hopefully give you a nice overview of this tutorial so that you can practice and look over it at your own time at home or in the workplace. Okay. So the key link you should have gotten is already is the GitHub link. So I just put it in the chat box here. Okay, so the Git so all the code and data materials are in this GitHub, and we also loaded this tutorial in Google Colab. So if you scroll down in this GitHub, you have a QR code which you can scan, or this link here that will take you to the Colab for this tutorial, so that you can follow along the examples as we go through them. Okay. Good. So everyone good so far? Is everyone able to navigate to the Colab? Say yes in the chat or give me a thumbs up. Okay, great. Okay. So yeah, so you can also use the emoticons to give any thumbs up. So that's great. Yeah, also angry faces also are fine if you feel like we're going too fast. So here's our kind of affiliations again. Generally just has background. We're kind of a data science group. We develop and apply machine learning tools to a variety of biomedical problems, mainly in the space of disease diagnosis and treatment. So we work with a lot with population cohorts that have multi-omics data where we can predict disease, onset disease, progression of disease, and in this case, drug response. Okay. But in this tutorial, we'll start off with a bit background on this problem, uh, so that you can kind of understand, what's the motivation behind this prediction problem. You also be able to understand kind of what currently what are kind of the most useful data sets for developing machine learning methods for tackling this prediction problem. Okay. In particular, the data sets include some of the omics data from cell lines and also some of the chemical data from that's extracted from the drugs, the compounds that represent the treatments, right? And we'll go through how we process that data, create feature embeddings from that data that can be used for machine learning approaches. And then a particular approach we'll introduce today is using graph neural networks to integrate these omics and drug features to predict drug response. So we go through this example, and it's not the only example for predicting drug response, but hopefully it'll give you a footing to look at multiple different approaches, different use cases in this prediction problem. Okay, so I'll jump into first kind of the general motivation for applying these machine learning models and tackling these prediction problems. And it's mainly to tackle two of the main obstacles in the drug development process. That being that it's taking very long. It takes very long to develop a drug, and it also costs a lot to develop a drug, right? That is because you start off with quite a large number of possible compounds that can be used as treatments for a disease, and then you have to kind of put it through a funnel and narrow down the possible compounds that could be potentially approved one day for treatment in human patients. So that involves a lot of preclinical assessment of drugs and clinical assessment, meaning testing in patients in clinical trials, phase 1, 2, 3, right? And as you move through this process, it gets more and more expensive for the drug developer, like a pharmaceutical company, right? So you want an approach that can identify at an earlier stage, right, before you commit too much money to testing, whether that drug is more likely to be efficacious in a patient. Okay, so that's kind of the main objective of our machine learning problem here, is to basically reduce the number of compounds that are under consideration, to try to identify compounds that are more likely to succeed, therefore shortening the development process, right? Because you have less drugs to test, and it should also save cost as well. Okay. So in this use case, in this particular tutorial, we're going to discuss about predicting drug response preclinically, so not in patients, in the lab, and particularly on cancer cell lines. Okay. So we're looking at drugs that can potentially kill cancer cells, right? And these cells are usually cultured in a petri dish in the lab, right? And we'll treat it with different concentrations of the drug, and then see how many cells, cancer cells, are still alive. So that's plotted. That can be plotted in a curve called a dose response curve, right? So at increasing concentration of the drug, we can plot out how many cancer cells are alive right after treatment. Okay. And so this gives, in ideal case, this gives kind of a sigmoidal type of curve, right? And there's some summary metrics you can extract from this curve, like the IC50 or the concentration at which you get 50% if efficacy in inhibiting the growth of the cancer cell. Okay. And there's also other metrics you can extract, like area under this curve, right? Or where this curve plateaus and so forth, right? So, so basically, in order to assess the overall efficacy of a drug on in this particular setup, on a cancer cell line, you would look at the curve and determine whether that curve shows a response or not. Okay. So basically, if the curve dips down quickly, right? So at even a small concentration of the drug, you can get quite substantial death of the cancer cell. That means we classify that as kind of sensitive response, right? And if it's like, there's really not much death in the cancer cells, that's no response, right? So, and the aim is to actually, have as much efficacy as possible at a lower dose of the drug, right? Because any drug, any kind of foreign compound you input has some level of toxicity, right? So you want to have the patients obviously take as little drug as possible, so to reduce the chance of side effects or toxicity. So therefore, we're looking for a curve that kind of drops quickly. So either characterized by a low IC50 or kind of like a low area under this curve, or even some of the shape of the curve can be deterministic of the response to the drug, right? But often the case, many compounds have this kind of the purple kind of response, right? It's somewhere in between the sensitive and the non-response case, right? Some drugs maybe have some response, when you get to a certain concentration, and it's a little bit difficult to say then, or a little bit more risky to go to patients, right? Because you're a bit unsure about whether that concentration, while it gives efficacy, whether it may also cause some unwanted side effects as well. Okay. So just to say, it's not really clear cut. It's not really a binary decision whether it's response or non-response responder or sensitive. A lot of cases are kind of in between here. Okay, so then what's the main prediction task is that you have a large chemical library of compounds, and this is true for, like, say any given biotech or pharmaceutical company. There's usually a library of compounds they're considering uh, uh, as potentially um, hitting a drug target, right? So a drug target is usually a biological molecule that's involved, say, a protein that's involved in driving the disease, right? And so we're asking the question, okay, first, because that drug hit that compound, does that drug hit that target protein, right? Does it induce any toxicity? May the drug also hit other proteins that are maybe not involved in the disease, right? Um, also whether that drug may actually get absorbed in the body. So even though you're giving the drug, say, to a patient at a certain concentration, maybe it gets metabolized by the body, so it actually doesn't hit the diseased protein, right? So there's kind of a lot of factors for determining whether a drug would give you an efficacious response, right? So instead of kind of going through all of these possible criteria, what usually happens is kind of a more empirical approach, whereby they just take a lot of drugs, a lot of different cancer cell lines, and screen them, right? So that's kind of the high throughput screening approach, where we're just taking a lot of compounds and putting them on cancer cell lines and seeing what potentially works, right? And not only on cell lines now, preclinically, there could be also organoids, or animal models that more model the whole environment, microenvironment of the diseased tissue, that models all the organs that might have an impact on the drug, say on the metabolism of the drug and so forth, right? But the data is generally generated, the data we're basically learning from is generally generated from these high throughput screens, whereby we're taking a large library of compounds and testing them on different types of cells. Okay, and so now to give you a bit more concrete detail about the data that will be used, I'll pass it on to Evelyn. We'll go through kind of first the setup of the environment, and also then the data we'll use.
Right.
Okay, now.
Okay. Hi, everyone. So I believe like with Danny's introduction, you have now have an understanding on the motivation behind of doing this drug response prediction and also the learning objectives of this tutorial. Yeah, so building on that, I will walk you through how to extract the data set that we will be using, which includes like the cell line features that we'll be using, and also the compounds features, like for example, the molecular structures, and also the drug response data that we need for later. So we can get started here. If you run this code there, it's kind of like help you to set up the environment. So it will do, it will clone the GitHub repository that, and also install some necessary dependencies here. So this will contain all the materials that we will be using throughout the tutorial, and all the codes that you need to, so that you can follow along the hands-on exercise that we have. I'll give you some time to do the installation and all that.
Feel free to ask any questions if you're waiting as well.
Yeah. Or if you have any problem running this. Should I continue? Or just let me know.
Yeah, I think you can continue.
Okay, okay. So for the next probably 10 minutes or so, so I'll be introducing about the GDSC dataset that we'll be using for this tutorial. So I also will highlight some of the publicly like accessible omics data, which you can find on the GDSC database. But today we'll be focusing on the specific types that we'll be working on. And then I will guide through some of the data processing steps to prepare the different data sets for the downstream modeling later. So if you can scroll down, Dennis. So the GDSC data sets here, they have quite a comprehensive landscape of drug responses. So this is across about 1,000 human cancer cell lines. And this data are basically being released in 2 different phases. So the initial phase, GDSC1, they actually screen about 403 compounds being screened in GDSC1 and also about 297 in GDSC2. So one of the key strengths of these resources is like they have the genomics data and also the transcriptomic profiles of these cancer cell lines, which been characterized through the COSMIC project. It's like a cancer cell line project. And for this tutorial, we'll be focusing on the GDSC2, which is like a more up-to-date kind of drug response data set. So before we dive into that, we can get started. If you run the code cell here, this cell here, it will import some of the essential libraries and also we'll download the data from the provided URLs here, you can see there. So at this point, after you have completed the downloading, basically you have all the necessary data ready for analysis in your local environment. If you can scroll down, there's basically you get some GDSC data there with SMILE structure, and also you have the gene expression data, as you can see. If you scroll down a little bit, you can see there's also the IC50, which is the drug response, one of the metrics that we'll be using. Okay. So now,
Did you have any questions? Or I'm
yeah.
not sure if.
Let me know if you have any question. It's just basically loading all the data. Yeah.
Hi, can you hear me?
Yes.
Perfect. I don't have any question. I just wanted to see if my mic is working. It is now. Thank you.
Okay. So now we'll go through briefly about the GDSC data set that you have loaded as well. So basically this data set, we have several cancer cell lines. So each cancer cell lines is being identified by a very unique COSMIC ID, and they are being categorized according to their TCGA cancer types. So these data sets, we have different cell line features. There's like mutations and copy number variation, and also DNA methylation, and also included the gene expression profiles. This can be used in any of the prediction, but for this tutorial, we'll be only using the gene expression data as like a cell line features. And then we will find that we'll be using some of the drug features as well. So basically, it's very important because some of the drug molecular structures are very key to targeting the proteins involved. So here we'll be using the SMILES, which is kind of molecular descriptors for that. And also we are going to use the compounds that is going to, which the targeting pathway that this compound is going to targeting to, which is by integrating both the cell line features and the drug features, now we can predict how cancer cell lines with different molecular profiles, they may respond to specific drug treatments, which then can impact the cell viability. But then how can we assess that? So normally drug response assays are used to evaluate the effect of the drug of a population cells across various concentrations, like Dennis mentioned previously. So the GDSC dataset actually include IC50, which is one of the key metrics, represent the concentration at which the drug reduces the cell viability by half. So a lower IC50 indicates that the cell line is more sensitive to certain compounds. And yeah, so we'll be using this as the primary dose response metric in this tutorial. So now I would like you to run the following code here. So this will process the GDSC2 dataset, and then it will group the cancer cell lines by their TGA cancer type. So once you have run this, if you can scroll down, the output will tell you how many cancer types are being represented in the GDSC2 dataset with the cell lines. If you can see here, for example, there's like 62 number of unique cell lines. They actually belong to the lung adenocarcinoma cancer type. And it just gives you a brief idea of the cancer type of these cancer cell lines being represented. So if you scroll down to the next one, we'll be loading the gene expression data here. So this is something that you have downloaded earlier from the URL. So this is like the basal expression profiles for all the cell lines. And here you can get a preview of the structure and contents of the gene expression data. So the first row here is actually the cell line identifier, which is the cosmic ID. And then the values there is their corresponding different gene expression level across, basically expression level across different genes. So each column represents like each different genes there. So if you run the next one, it is when we can normalize the gene expression data. So basically, this is to ensure that all gene expression values are on the same scale so that they are actually comparable across different cell lines. So basically, this is to transform it into a more standardized format, making it more suitable for the downstream modeling. And you can see it's actually each gene here, you're looking at its expression across all cell lines. And this is being mapped to each cosmic ID. And once you have normalized the gene expression data, they are ready to be used as one of the features for downstream. Let me know if you have any question regarding this. If not, I'll move on. So next, as I mentioned earlier, so there are like different parts, right? Yeah.
So I see we have what is it, five different rows here? Are those 5 different cell lines or cell types?
Yes, so this is just a preview of the top 5 rows. So
Okay.
You can see like each row is each cell line, and across that is their different gene expression of that particular cell line of each row.
Okay, perfect. Thank you.
Yeah. So yeah, so we have done with the cell line features that we are going to use for this tutorial. So next we'll be using, we're focusing on the drug features that we are going to use. So previously we have extracted drug features in the GDSC, which is first one is the molecular structures, which will be you focus on the SMARS, which is like a string-based representation for molecules from the QSAR modeling. And then, so this is one of them. And another one that we'll be using the drug features is the biological pathways that the drugs target. So just briefly, like pathway represent the biological processes or like the signaling pathway that these drugs aim to inhibit, so that it will kill this cancer cells from, or like cancer progression, so that the cell like stop growing. If you scroll down here, you can run this cell here, and you will see there's a list of pathway targeted by drugs being screened in GDSC2. So here, this kind of helps us to understand a little bit of the range of biological mechanism that are being targeted by the drugs in GDSC2 dataset. So yep, so you have everything here now. So we have, after loading the gene expression and the drug features dataset, we will now move on to the drug response data. So this has been loaded previously. As if you can run it, run this down here, you will see the IC value. So we'll be using the IC50 values that were provided by the GDSC database. So like I mentioned, this value allows us to evaluate how sensitive is it of these different cancer cell lines to various drugs, and also to predict how effective this drug might be in killing or inhibiting the cancer cell viability. So if you scroll down, then if you run this code, as you can see, this is a preview of the drug response data here. The first column is the PubChem ID, which is the column contains like the unique identifier assigned to each drug in the PubChem database. And then the second column, like I mentioned, is the cosmic ID of each cell line. And then from there, you can see, so this drug screen against this cell line, what is the IC50? So you can see here, there's natural log IC50 values. So yeah, that's you're all set because you have all the cellular features. We have loaded the drug chemical features already, and we have the drug response. So later, Yurei will walk you through how we can using these features and the drug response data set, and then apply in the deep learning approach to predict drug response. Yeah, that's from that data processing part. Let me know if you have any question before Yurei, before I pass on to Yurei.
Thank you. I just want to make sure I've understood everything we've done so far. So we've collected the IC50. Did I hear you correctly? We've also got the structural data of the drugs.
So it's actually in the first, if you go back to the first, yeah, here you go. The GDSC, and then you scroll down a little bit, and then the smiles features is actually in one of those. Yeah, here you go. Here you can see the smiles. Yeah, it's actually there.
Are we using the smiles data?
Yes.
Okay.
Yeah. Yurei will explain further on how it will be used later on.
Perfect.
Yeah. Any more questions on this part?
Yurei, would you like to share the screen for the next part?
Thanks, Evelyn and Dennis. Oh, I thought we still have one question from the audience. Because I saw there's a hello to Evelyn.
Oh, right. Yeah.
How to interpret the final result? I think I share the screen first, then. Hi, Ruben. May I know like what's final results you are referring to? You can either type or maybe you join the audio. You mean here, this table? Okay, so maybe I illustrate from here. So for the drug response prediction problem, we apply a drug to like some cancer cell lines. So we have like two items, right? So for the PubChem, is actually the indicator for the small compounds, for small molecules such as drugs, while the cosmic is representing the cancer cell lines. So this draw meaning that we apply this drug to this cell line, and the IC50 value, which after the log transformation should be this number. So while the final column representing like the sensitivity of the drug to the certain cancer cell lines. Does it make sense to you? It's clear to you? So the cosmic ID meaning that is like the identifier to the cell lines, while the PubChem ID is identifier for the drugs. I can illustrate a bit more on the details of how we get the data. So if you go to the folder here, right? So we can see we already git clone the total projects under your root. And you can see there's a data folder that's based on the natural log. Yes. I think it's a log2 value, right? I think it's it's I think it's based on the natural log value, not the log2. The natural log value, yes. So you can see we already downloaded the data like for the following data tables. Okay, so that's from using this class I predefined, and you can find the user functions here in the GitHub repo. There's a utils function, so this GSC processor helps you download and process all the data. And if the data is already loaded at your site, so you directly load the data. But if you want to like run the total process and download the data on yourself, you just delete everything under this data folder and rerun this these cells. So it gives you like directly from the very beginning how the data is downloaded and how it's loaded. Okay. So if you want to know like more detail in the coding, you can check this utils file, and every function is defined here. And I think that's it. Do we still have more questions to the previous section? Okay, so I just continue and stop me like whenever you have a question. And as Evelyn and Dennis introduced before, we know the settings for the machine learning for drug responses, and we know like what kind of data we are trying to use. So drug actually a supervised learning problem where we're trying to mapping the input feature spaces to the output spaces. And our input feature spaces, and as I introduced here, right, we have the drugs and also the cancer cell lines. So normally the drugs can describe by the chemical properties or its chemical structures information. And for the cell lines, so it is described like in several ways. For example, the multi-omics features, or like where the primal cancer tumors it comes from. So in the deep learning settings, so we're trying to build a non-linear function to mapping, like learning this complex relationship between the input features to the outcomes. And in this section, I will try to illustrate what kind of data we are using and the key components in the drug response prediction problem. For the drug features in this notebook, we will use structural information for the small molecules, where the small molecule is represented in the graph as a graph. Each of the atoms here can considered as a node, while the chemical bond is considered as an edge or the connection formation in the graph. And we use a gene expression value to represent cancer cell lines features. So normally we have like more choices. For example, for the drugs, we can have is like more chemical descriptors, or even it's called the fingerprints, while it encodes the substructure. And for the cell lines, we have more omics profiles, for example, the mutations, genomics, right? And we have like the copy number variations for us to use. But in this project is more like try to do in a tutorial, we're focusing on representation learning for the graph structure, and also use a gene expression to represent cancer cell lines. So normally, there are three main components in the deep learning models for predicting the cancer drug response. First of all, it's called a drug encoder, where we're trying to map the initial input space for the drug. Right now, it's a molecule graph via this graph neural network, and to the hidden space. And then for the gene expression values, we have the cell line encoders, which also like kind of the data compression process to mapping the very high dimensional gene expression values to a latent space. And afterwards, we have the latent representation for the drugs and cell lines. We map it to the final prediction subnetworks and get our drug response prediction score, which is measured by the IC50 value. And since it's a supervised learning problem, our goal is to like learning the parameters in these three components by the optimization functions, right? So before like everything is settled, we need to split the data first. Okay, so our data is represented in this way. Like we have drugs and we have cell lines, where the pair of it has a IC50 value, which measures the efficacy or sensitivity of the drug to a certain cell lines. So we can also represent it in a data. Oh, sorry, I just want to qualify. Oh, it's from Dennis. Sorry, I thought there's another question. So here is another representation for the drug response value, where each row is a cancer cell line and each column is a drug applied to the cancer cell lines. So the values inside is IC50, where the drug is treated on a certain cancer cell lines, IC50 values, and we need to mask some of the certain indices in this matrix so that we make sure the certain pairs of the drugs and cell lines combination is not seen in the testing set versus the training pairs, right? So in these examples here, all the pink cells are our testing samples, while we train the model on the remaining like the numbers here, right? So we're training the model, getting the parameter tunings here, and we make the prediction and testing the model on this these blocks. So here is called a mixed test, where like all the cells and cancer cell lines information are already seen in the training site, while we have like more difficult scenarios. For example, we mask all the testing set is from like these two columns. So meaning that these two drugs information are not seen in the training set. Or we have like another kind of, it's called a cell line masked prediction. So this cell line information is not seen in the training set. So different training and testing splitting has different purposes. But right now we use this mixed test, which is like a more general approaches to illustrate how we train and test in the model. So afterwards, we have the like training. We have split training and testing. We will optimize the parameters here in these models, and we make the prediction of the drug responses on these cells and test the model performances. Do you have any questions here? So afterwards, I will like illustrate how we process the drug features and how we build the neural networks, including the graph neural network for the drug encoders. Okay, cool. So in this tutorial, for the following sections, especially 4 and 5, we focusing on the graph representation learning, especially for the small molecules. So the structural information of the small molecule, the drugs, is quite important, like in determining its chemical properties, right? For example, like the drug needs to bind a protein, then like influence protein networks to finally showing some like biological relevance to the cell lines. So the certain substructures can influence the binding affinities of the small molecule, and also other key properties of the drugs, for example, the solubility and even the toxicity, can reflect it by its like essential properties, which is determined by either the total structure of the small molecule or its motif, which means like certain substructure of these graphs. So in the cheminformatics, this SMALS, which is a simplified molecular input line entry system, is used as a notation for describing the structure of the chemicals compounds. And here is a certain example we will use to to illustrate how this like smiles can represent a drug, and how we transfer it like to a chemical graph, and how we map it like to a really mathematical graph and use a graph neural network to process it. So the key steps in this section have like totally 3 steps. First of all, we need some like, we need some like toolings to transfer this mouse representation to molecular objects for this molecule. So we use RDK packages in Python. And afterwards, we need a graph representation, which is represented like a mass formula for this mouse. So what is actually the small molecule graph? Okay, and we will visualize what we have after transfer the smiles back to the graph structure. So I will starting with this compound information has a smiles in this formula. And at the very first step, we can visualize, we can run these cells. Oh, and let me know if you get any like import error or cannot run the cell, because like RDK is a bit fragile on the Colab. If not, I will continue. So you may see like this figure. You can see we directly generated this. It's called a molecular objects in the RDK packages. And so the atoms here, so the nodes here, which is not represented, is a carbon atom. Well, here is oxygen and nitrogen, right? So the other hydrogen are like already being ignored in the figure representation. And why we can call it as a graph? So mathematically speaking, graph is like a set of nodes that are connected with each other, where the connection information can be documented using either it's called an adjacency matrix, or we call this edge list, meaning that if it's documented by if these 2 nodes are connected or not. Okay, so if we want to transfer this small molecule to a graph objects, we need to consider each of the atom as a node, whereas certain chemical connections as edge information. So we ignore it's a 2 bonds or just a single bond here. We can draw a graph connection formation. Okay, so we still keep the labels here for each of the node. So it's still a carbon, right? So, but we ignore like the different colors. Can you run it properly, this cell? Okay. Yeah. So to define a graph mathematically, we need like, especially this molecule graph, we need like two sets of information. First of all, what are the nodes, right? So because the nodes here we use are the different atoms, but different atoms has different meanings, right? So it's a carbon atom, it's an oxygen, and it's a nitrogen. And we need to document this information first, and we call it as a node feature. It's an initial representation for the small molecule graph. And there are different type of features for atom. So here in this project, we use a very simple representation to just distinguish different atoms. So we directly use the atomic number, right? So the carbon has atomic number 6, and the oxygen has 8. But there are other informations we can use. For example, we can consider does this atom in a ring, or its degree, even the formal charge, right? So this can be used as a atom level information. But in this project, we just try to distinguish different atoms. So we use atomic number directly. So let's run this graph first, and I will introduce the other information, which is also important. We need to document the connection information for different nodes, right? Because here we know this carbon is connected to 1 oxygen and 2 nitrogen, but it's not considered to the other carbons. So we need to like map them to the certain, it's called the node indexes, and we document the connection information by it's called either edge list or the adjacency matrix. Okay, so it's what we map in our from the previous graph small molecule to the mathematical speaking is a molecular graph, and this index is how we like documents the node positions. There's no meaning for it. We can like do the perturbation on all the informations, but after we do the perturbations, the adjacent matrix or the node information, all the connection informations also be changed, but graph structure remain unchanged because we just know this node, it has like two neighbors, has three neighbors and connected by the edges, right? So different colors here in the graph representing different atoms. For example, this blue one means it's a carbon atom, and the red ones are the oxygens. And for the node feature level, it's an atomic number, and the carbon atom has atomic number 6, while the oxygen has atomic number 8, so on and so forth. So as we already have documented this, like written down the information for these nodes, we can also write down the information for how these nodes are connected, right? So we know this node 1 is connected to 2, 0, and 8, and it's not connected to 4. But this 5 connected to 4, 7, and 6, but not connected to either 1 or 3, right? So we have two different methods to document this kind of information. The first one is called the adjacency matrix. So it's a binary encoding where here we have like 1, 2, 3, 4, 5, 7, 8, 9 nodes, right? So it will be a 9 times 9 matrix where for the indices in the adjacent matrix I and j, it means that these ij nodes are connected. It will be 1. Otherwise, it will be 0. So if you're trying to run the code here, you can see our example of the edge representation. So another equivalent documentation way is called the edge list representation. So other than store like a more largely adjacency matrix, which will be 9 times 9 in these cases, we can directly document all the possible edges, which is like documented in a pair, right? So for example here, the undirected edges, meaning that this 1, 0 are connected, while 0 and 1 are also connected. Because there's another directed graph, but we are not using here, as we assume that how the information is like not following any direction in our small molecule graphs. And here is our example for the adjacency matrix. We can see it's a 9 times 9 matrix, where the 1 here, meaning that it's in the first row and the second columns, right? But in Python, note that the indices starting from 0. So meaning that it's a 0 by 0, 0 times 0, 0.0, and this is a row 0, column 1. So the 1 here defines the connection here between the nodes 0 and 1. While if we looked into other informations, for example, it's a 8th row and 8, 7, 6, 5. The 8th row and 5 columns, meaning that there's a connection between 8 and 5. Do we have it? Okay. Should be, oh, sorry, it should be 7 and 5. Yeah, so the edge here. Sorry, because it's from 0 to 8, not 1 to 9. I mistake. So it's like the 7 row and column 5. So indicating the connection here. Right. Anyone like following me? If you got any questions, you can just stop me. Okay, so with the feature information and also this connection information, we already can define a graph, right? So it means that if we have the adjacent matrix here, or if we have the undirected edge list here, one of them is enough. And with another information is called like the node level features, we can reconstruction this graph. So it has equivalent information. Okay, so that will be the input features for the graph neural networks. And for the following step, we need to do this kind of processing to all of our drugs, because we have like 100 drugs in the data set. We can use similar ways to process all the drugs here. And you can see in our drugs, what we have the like different information. For example, the drug ID is the internal ID used in the GDSA data set, and we have the drug name. So it's like the small molecule. It has different like synonyms or its name representations, right? And in the GDSA data set, we also know its primary targets for these drugs, because these drugs are already like tested on a lot of cancer cell lines and even the targeted cancer pathways. We are not using this information in training the data, but yeah, it's definitely useful for developing your own customized machine learning methods. And the PubChem ID is what we use to identify the different drugs, and GDSA2 is the data resources. And we can check that like for different drugs, different number of cell lines are tested, because like if we come back to here, the example, we can see that there are some like blank values here. It means that the drugs is not screened on that cancer cell lines. Okay, and finally, the SMILES information is what we will start. First, we transfer this SMILES information to the RDK molecule objects. Then we process it with our customized codes to make it in a small molecule graph, where we generate atom nodes information and also the connection informations. So we just need to iterate over this data frame. And finally, our processor data looks like this one. So this PubChem ID is identifier for a drug, where the initial point starting from the SMILES information, and we get this different atoms, right? So this different number means the atomic number for the atom. Remember, the 6 for the carbon, while the 8 is for the oxygen. And these two columns are the adjacency matrix or the edge list, which is equivalent. We can use either of it, like for different input of the models. So it documents like the connection information in the graph. And finally, we can use this torch geometric data to create the data objects. Okay, so torch geometric is normally used in the graph machine learning and graph neural network. It has a like backbone of a lot of popular models, for example, convolution neural networks, graph attention neural networks, and also this predefined data classes for us to store our data like in a more widely used format. Okay, so after we process drug features like into the ways we needed to use to build the machine learning models, we can finally prepare the whole molecule graph dataset for the deep learning. And as introduced before, we use 70% of the data for training and the others for testing. So if you want to like train for other portion of the data, you can edit it here, like a test size, right? And also here, the random state is to confirm that a notebook is reproducible, and you can change to other numbers and to see the performance is stable or not for the training and testing. And for this drug response data set is I predefined here. I think you can find it also in these utils functions. And here are the details, but I don't want to go into details. But the output should be the cell line level features, which is normalized gene expression values, and the drug features, which is like the graph objective for the small molecule. And the IC50 value is our prediction goal, right? So it represents drug responses. So here we directly run this, and we get our training and testing set. So here we get like over 100,000 training samples and 4,600 testing samples. Then for the model training of the deep learning, normally we need to patch the data in small batches, right? Because we have so many data points, and also the model has a lot of parameters inside. So it is not possible or computational inefficient to calculate the gradients from loss function using directly all the samples. So normally we do is called a stochastic gradient descent based on small subsets of the feature of the training sets. So here, for a batch of the data, it's meaning just a small subset of the training samples. So in each of the samples, here I define like using 2048 samples. You can use either smaller or larger values. It depending on like your computational resources, right? If your computational resources is more powerful, you'll have like more GPU storages. You can like increase it, make the normally the larger batch size will help you to get like more stable training stages. So here is example batches in our loader. So this tensor, which is actually a matrix, is the gene expression value. So each row is still the gene expression value for this cancer cell line. Well, this data batch is our graph, is our like chemical graphs, right? So it is stored in a large graph, while then the graph will also be cut into small batches. So the final tensor will be the IC50 values. So these numbers are this drug on one of this this cell line on one of this drug gets an IC50 value. Okay, so each of the indexes is mapped. Okay, so that's everything for pre-processing the small molecules data from scratch and how we handle the deep learning model data process. Do you have any questions here? Let me know. Yeah, we can go to like even more details into our code space. Yeah. If no questions, I will continue to the graph neural network parts. And if you get any troubles in running the code or understanding, just let me know. Okay, cool. As we already have the small molecule graph, we can use a graph neural network to process the data. Like here, remember we have a drug encoder, right? So we need to map this input drug informations to a hidden space, which is quite relevant to our prediction goal. So this graph neural network will be used here as a drug encoder. Okay, so normally graph level supervised learning is referring to the classification or the prediction result on using the entire graph structure, because there are other tasks for graph neural networks. For example, on each node, there's a node level information. For example, like the recommendation system, right? And also there are some edge level tasks. For example, if we have a protein interactions, we want to predict after we got some new proteins, do they have interaction edge listed information to the existing proteins? But in our in this project, the graph neural network is referring to it's called a graph level task, where we need to encode the drug small molecule graph to a latent space and doing the final prediction result. So here in this section, the learning objective is trying to understand the basic settings of the graph neural networks, and I will let you go through like two general approaches, which the one is graph convolutional neural networks, and another one is called the graph isomorphic neural networks, which is quite popular in the small molecule representation learning. And a quick catch up is what we have already a feature matrix, right? So that's for the that's for a small molecule. If mathematically speaking, a feature X has capital N nodes where we have like N atoms here. Like in the previous examples, we have 9 atoms, right? So the number of features is right now we only use atomic numbers, so this D is only equal to 1, right? So we have a 9 times 1 matrix for the previous example drug. So normally it will be a N times D matrix. Then we have a adjacency matrix where it's N times N, representing the connection information in the drugs. So our goal during the training is we want to get the graph level representation for the small molecules. But how do we achieve this? We need to first learn it's called the node level representation, where I call it a new feature, which is like more interpretable during the training process. So we map this N times D input matrix to the N times F. So normally F is like the hidden dimensional spaces, right? So for each of the node in the small molecule graph, it will get a new feature which has F dimensions. And after that, after we get like the novel node level new features Z, we do is called the readout or the pooling operator from the to the graph neural network. The pooling operator should be the perturbation invariant. So normally we do the max or the mean pooling. So mean pooling meaning that we take the average among all the atoms to get a 1 times F vector to represent a graph. Okay, it's similar to the max pooling function, where we take the maximum among all the rows and get a 1 times F vector for the graph. Okay, so the graph neural network is defined by the stack of different hidden layers, while the most popular schemes is called the message passing graph neural networks. So what is really the message passing here, right? So normally we call the information initially defined on each of the node is called the messages. Well, the message passing is that the node obtaining or updated the information from its direct neighborhood. So for example, this 1 and 2 are connected, right? So it has a direct neighbor of 2 for this 1 node. So it gets information from the 2 from the first step, right? But this 2 is more like a hub node. So it is connected to all the other nodes. So especially, so except for itself, it receives the messages from 1, 3, and 4. So finally, it has like more comprehensive messages received here. So similar informations for 3 and 4, because it has 2 neighbors. So you can see like 2 more messages are received for these 2 nodes. Okay, and one simple message passing graph neural network is defined by the first of all, this A times H. Normally we can do the very similar stuffs for achieving this, right? So it's a summation of all the newly income messages, but this this W, meaning that, so we do not directly we do not sum them up, we aggregate them by like a weighted matrix, right? So it's like a weighted sum of the information received from the other neighbors to a single node. But there are some certain limitations for these definitions. For example, if we consider this node 2 is like a hub to this graph, so it definitely receives like more messages from its neighborhoods, but the 1, it gets only like information from 2. So if we consider like a more extreme cases, we have like many nodes, but only one of them are connected to all the others, but the others only connected to the hub nodes. So except the hub nodes are receiving all the information from its neighborhoods, all the other nodes are only receiving piece of the information from the hub. So meaning that if the graph is not normalized, so the information will get like not even distributed on the graph. right? So that gives us an iteration to design is first called the graph convolutional neural network. The graph neural network, a graph convolutional neural network, trying to address the limitation described by this way. So first of all, it adding the self loop to make the adjacency matrix more stable, and then normalize the degree on each of the node, meaning that so here is a normalized adjacency matrix. So meaning that we don't want this this hub node receive like very heavy informations, but the distribution of the messages on the graph is more even distributed, right? So this updating scheme is what we call the graph convolutional neural networks layer, and this delta function here is called the activation function, where that's how the nonlinearity in the neural networks comes from. Because like here, it's only like the linear combinations of all the weights, right? But after the applying the activation function, for example, the reloop function here, and we stack them like in more different neural networks, the nonlinearity from the feature spaces are added onto to the hidden spaces. So in the following part, we will go through like how we implement graph convolutional layers from scratch. I know that we can also directly call the functions from the touch geometric, but in this tutorial, I will lead you like build calculate it by hand. So it's not very like complicated. We can directly calculate all the informations here, right? So considering what we need actually to do the graph convolutional operator. First of all, this edge at the initial state. So this edge L+1, meaning that the hidden state has a L+1 layer. Well, this edge is like before the graph convolutional operator is applied. So if L equal to 0, it becomes to our input feature, which is like which is here, right? So this is H0, the different atomic numbers. So then what we have this A here. First of all, this A is a adjacency matrix, and we add in the self loop. Self loop meaning that each of the node is connected to itself. So it's called a self loop. So this A tilde is actually the A plus identical matrix. And this D is just a normalization of the adjacency matrix. Okay, so we can directly compute this D tilde from the A tilde. And so with the A and the H, we can directly compute a graph convolutional operator. So at the first step, we added this like the self loop to the original adjacency matrix. You can see here, before that adjacency matrix have like all zeros because each of the node don't connect to itself, right? Then we add it's called the identical matrix here, and you can see like the diagnosis, the diagonal entries of the adjacency matrix become to 1. And then we trying to normalize adjacency matrix where it's calculated by this way. And you can see after that, all the information on the adjacent matrix are already like normalized. And so this W is what we're trying to learn from the model, but from the initial step, we need to get a random initialization for it. And afterwards, like during the model optimization steps, it will be like finally learned to map the input features to the final outcomes. And also we need to define the non-activation function, non-linear function of the activation functions, where we use a reloop function here. And also we have other choices, for example, the softmax, right? So our graph convolutional neural networks are stacked by this. So initially this edge will times weight matrix W, and also the normalized adjacency matrix A tilde, and afterwards we added in this activation function outside this multiplication. So that's like a more detailed implement. We take an example here. We use an output dimension where output dimension referring to the this f of the description, where it's a dimensionality of the hidden space, right? So we can either increase or decrease it to add in more complexity in the model or reduce it. Okay, so the initially this small molecule has 38 atoms and only one like input feature, right, the atomic number. But after the apply the graph neural networks, we get it like into the 4-dimensional, because it's also need to do the pooling at a final graph level output. Okay, so for this GCN output, we get a node feature after the graph convolutional layer. It still keeps the number of nodes, but we mapping the initial features from the 1 to this hidden dimensionality 4. Okay, do you have questions here? Because like this implementation is more like a bit more detailed. So just follows equations here, trying to understand and get an intuition from here, trying to understand like what we are doing. So it's like the re-implementation, you can get like the exactly same output calling the touch geometric functions. Okay, but what I want to show here is that we can actually also building from scratch. And the next step should be the pooling functions for the graph level task, because for now we still get like for different small molecule graphs, we still get different matrices, right? Because we have different number of nodes. For example, here, this molecule has 38 atoms, but the other one, like we showed before, has only 9 nodes. So how we want to like map them into a same space, especially in the same shape. So we need to do this called a pooling function for the graph level. So we need to reduce this 38 times 4 to a 1 times 4. So normally we either apply it's called the graph mean pooling function, where all the nodes information just take the average, or it's called the global max pooling function. Right, so it means that like for each of the dimensionality in the hidden space, we take the maximum number of it. And finally, so right now what we are implementing is like the stacked graph convolutional layers inside these hidden layers, but we need to make it like deeper to gain more message passing on the graphs. So after stacking them like into a few layers, we can get like a final output. So the structure of the graph keeps still as an input, but all the representation on the nodes are already updated during the message passing. So graph convolutional neural network is trying to solve the problem where like the hub nodes get too many connections to the other nodes, while this graph isomorphic network is trying to solve another great challenge in graph matching, where like it checks if two graphs are actually have like the same structure or not. Okay, so it's especially important in the graph representation learning for small molecules, because like different graphs with different structures and also different substructures, they have like different chemical properties. So this graph neural networks has a key components of, first of all, not as a so in the graph convolutional network, it's directly doing the linear combination of the messages on the normalized adjacency matrix. I'm sorry. Well, this graph isomorphic neural networks does not like doing this W functions inside. It doing a multi-layer perceptions outside this aggregation. So still the information is aggregated from one node to its neighborhood, but then the model weights and the nonlinearity is added outside like the GIN updating rules. Okay, so more details can be found in the paper, and it has like a nice illustration of how it proves the WL test. But in this same formula, it performs well in the graph representation learning in the small molecules. So in the following parts, we will use these two models, the graph convolutional neural networks and the graph isomorphic neural networks, as drug encoders to build the drug response prediction models and compare these performances. Right. And before going to the next section, that is more implementation for training the task. Do you have any problems or like harder to understanding parts in this section? Because there are like many questions here, maybe it's hard to following, but trying to like read the code and run right, like you can see the exact examples how we perform graph convolutional operators here. Okay, so as I already introduced, like some two types of the graph neural networks, we'll use them to as a drug encoder to build a final drug response prediction model. So two inputs for this project is one is a graph molecule graph. We will pass it to the graph neural network layers and get drug representations in the hidden space. And here, the original paper, they use genomics features, but right now we use gene expression values. So it won't be the binary encodes for the mutation on the genes. It will be the gene expression values, so discrete numbers. So still we feed them to the convolutional layers and finally get a cell line hidden representations. And then we feed them to another stack multilayer perceptions to build to predict the final drug response, which is measured by the IC50. So for more detailed of the training codes, you can find it here, still in the, I think still in this utils path. You can find all the training functions here, and also our testing functions here, right? So to save us more time to introduce notebooks, I won't run the training parts because it takes a while as we don't have a GPU on the Colab, but we can directly load the model weights. So the model weights is already uploaded to the Colab, so it's under this weight. You can see we have the, where is it? Okay, so you can see there's a GCN model and there's another GIN model, meaning that this is like the a drug encoder by the graph convolutional neural network, and it is a drug encoder by the graph isomorphic neural networks. Okay, so we directly load the model weights and trying to test the models. So what's the detail of other model? I will show the model GIN. Well, we have like 5 layers of the graph convolutional informations, and our initial embeddings is from the possible atomic numbers mapping to the hidden space and feed them to 5 layers of graph convolutional neural networks. And for the cell line branch, we have a stack of deep neural networks mapping from the initial feature spaces to the the hidden spaces. Finally, we feed the combined feature, which is 1 to 8 for drugs and also 1 to 8 for cell lines. Combine them together, feeding them to another few stacked layers, and doing the final prediction where we get drug responses, prediction values, the predicted IC50. And we test our model with predefined model weights. It takes a few minutes. Before that, we get a result. We can go through how we evaluate the model. And So as it is a prediction task, especially it's a regression task, we use like various evaluation matrices to test the model performance, if the model gets like accurate prediction or not. So the first of all is called the mean squared error, where it's a L2 loss actually, like how far the predicted value yi had as to the initial true value yi. And also the mean absolute value, which is actually the L1 loss here. So it's absolute differences in the prediction to the true values. And also the R square value is and the Pearson correlation. These two values are evaluating like how the predicted values and the true values are close to each other, like in the linear spaces, right? So if the PCC and this R square is near to 1, meaning that the prediction is better. But for this mean square error and the mean absolute error, if they are close to 0, meaning that the model gets like better performances. Okay, so yeah, after 1 minute, you can get your predictions and true values from the loaded model. So have you run the code and get exact same predictions as I have? If so, I will run the following cells and we can check how our model is predicted. So in this test DF, after we run the predictions, we can say the PubChem ID is for like it's for a drug, and this is for the cell line identifier, and it is a regional IC50 value, which is a drug response. And so here, these two values are like the predicted from the model GIN or predicted from the model GCN. You can see like from this few samples, looks like the values are close enough to our original values. But to evaluate like the model performance, we need to check the overall performance like on all the testing data points. And if we visualize the true versus predicted values for the drug response, these blue dots are from the graph isomorphic networks, and the orange part is from graph convolution neural networks. They get similar performances, but from this rooted mean square error metric seems graph isomorphic networks gets a bit better results compared to this graph convolution neural networks. And we can also check the performances like for each of the drugs, right? Because here is different data points are actually for drug cell line pairs. But what about like the average performance result on certain drugs as we build our model, mainly focusing on the graph representation learning on the small molecules. So let's run these few sections, and we take two examples here, which the one gets poor performances, where another drug gets like here performances. We can see, so here this drug like gets this carbon ring like more separately, well this drug has like different structures, and from the structural informations looks like this structural like is quite diverse. So it makes sense a model gets predict not well in the certain structural spaces, but it's better for the other spaces. And this performance also may due to the like the training set and testing set, maybe this kind of structure are more relevant or more similar to some certain drugs in the training sets. Well, this one is more like unseen drugs. Okay, so to see if our nonlinear and deep learning models really can boosting the performances, and if the model really gains some like incremental in performances from this graph structure and nonlinear relationships, we also build the soul regression model, which is a linear model with penalties, to see the like if without this kind of complex nonlinear informations, how the baseline model will looks like. And if our model really gains some information from the small molecule structure and the nonlinear relationships. So what we do here is we only use like the gene expression values, but the model is fixed per drug. Okay, and the result is aggregated for all the drugs. And so the graph structure information is not considered here in these linear models. We want to compare the performances between this lasso model and our graph neural networks. So we give it a few minutes for it to train and testing. I think normally it takes 2 or 3 minutes. And during the training and testing period, do you have any questions so far? Yeah, and I want to emphasize, except for this code lab, you may find the codes here in the GitHub where we define the models here in this graph, DRP GNN. Okay, so the DRP is short for the drug response prediction, and the GNN is short for the graph neural networks. So we have the DRP models, which takes either the graph isomorphic network or the graph convolution networks. And you can change the model structures here for you to play, right? So can define, like, for example, the embed dim. That's a hidden dimensionality in the spaces. And also you may want to try more like graph convolution type, like the graph attention neural networks. And yeah, and also we use let me check. Yeah, we use a graph add pooling function here, which is similar to the graph mean pooling, but it does not take the average, but directly takes the added for all the node informations. You can also try to change it to the graph maximum pooling functions to see the model performance changes. Okay, so we get the result for the linear cases machine learning, and so that's this scatter plots is for different drugs average performance on the testing sets, and the lower RMSE means better performances. So on the x-axis is a performance for the graph isomorphic networks, where the y-axis is for the lasso models. So the higher meaning that the model is like worse, right? So for most cases, we can see the GNN model outperforms the lasso model. So meaning that indicate that our nonlinear graph neural network models really learn some useful representations from our graph objective data compared to like the very baseline model. So in conclusion, in this tutorial, I and Dennis and Evelyn, we try to introduce machine learning for the drug development and drug responses, especially on the cancer cell lines, focusing on the graph representation learning for the small molecules, right? So we use the IC50 as a drug response measurements, and all the model is trained on like pan-cancer cell lines. Through the integration of this graphs representation and also gene expression data, we can illustrate how we build the deep learning models on this problem to capturing the nonlinear relationship between the input where the drug structure and cancer cell information to the drug responses. So from the notebooks, there are some like very detailed technician points you may want to follow later. First of all, the data processing part, as I introduced, you can delete all the files here and rerun the first cell. You can know how to download and processing the data from the GSA2 dataset. And we have all the codes for training and reproduce the models for graph neural networks. And we compare the GNN model with a simple linear lasso model. So it's also quite important during this kind of machine learning research. We always want to say if what part of our model really learns useful informations. and we need to compare with the previous model to see if our model is really effective or is because of like other issues. So we evaluated our model with different graph neural network models, for example, the GCN and the GIN, and also we compared it with Lasso models. So in conclusion, in short, using this graph structures information and the graph neural networks, it gets more potential in this drug development and discovery process, and maybe we can use it like in the other researches also. Okay, I think that's all parts from me. Do we have any questions from the audience? And Dennis, do you want to also like add some comments?
At the moment, any other questions? Sorry, it was a lot to go through. So don't worry. I'd say just don't worry if you didn't follow all of that. The point of this tutorial and providing you know the collab and the GitHub is to so that you can also go through this later on in your own time. GNNs are quite flexible as well. So in addition to modeling drugs, you can also use it to model relationships in the omics data, right? So in just like say like relationships between RNA, proteins, and its interactions as well. So and then this kind of approach can be also used to model any other types of not just dose response data as well, any other clinical outcomes or health related outcomes can also can also utilize this approach.
Yeah, I think we got 2 comments from the audience. So for the Cameron, yeah, thanks for attending it. Yeah, if you are interested in the GN models, actually have more tutorials in this GitHub. So you can go through. There are exceptions for this collab, right? It's called the APBJC tutorial collab. We have a long version for it, and also we have a building from the scratch for the graph neural networks. And you can even find like some more implementation of the from the scratch how we get graph convolutional neural networks in different ways in these files. Yeah, it is. I think it benefit for you to play around. And from Ruben's question, yeah, this is supervised learning. So because our final goal is if we trying to follow this illustration parts, our final goal is to predict the drug response value. So the loss function is actually measure the differences or the error parts from the predicted drug responses versus true drug responses. And all the parameters here in the encoders of the deep learning models are learned. So yeah, it's supervised learning. And also we get like more publications on the machine learning for the drug responses. Not only these two are like the using a graph neural network and the deep learning models, and we have another one to predict the whole dose response curve with uncertainty qualification. If the model performs better, it can be used to predict response for novel molecules. Yeah, so there's one particular scenario for this kind of novel molecule prediction. For example, here each row is a cancer cell line, and each column is drugs, right? So in this tutorial, in this collab, the training and testing are randomly split. But if we want to see the model performance on the novel drugs, normally we train on like the first few columns and we test them on the new drugs. Then we can test the model ability to predict the novel molecules. I hope this answers your question. So like different data training and testing split can give us different like evaluation for the model. So for the novel molecules property prediction, normally we train on certain molecules and we predict on the others.
So it's just the transpose of that. It's the you would leave out the rows because the rows are the compounds. Just the columns are the cell lines. So we would train on certain rows, like rows 1 to 5, and then you could also predict the response of drugs without and having seen any responses from that drug. So like a case of a novel compound. Yeah. And I just to add to that, I think like this may be where graph neural networks excel a bit better, excel more than other approaches, because the intuitions that the graph kind of models mirrors this kind of the interactions between the atoms within that compound, right? So that if you have a novel compound, you may not have seen that exact chemical structure before, but there may be some similar chemical structures with other known compounds, right? That you can learn from. Good. Is there any other references or other helpful tutorials you would suggest, Ray?
Okay. So
outside of the one you've given, like if there's any other.
Yeah, one particular I think is you can try to follow if you want to learn the graph neural network. I suggest you search the touch geometric, and it has a there's a list of there's a list of videos here from the very scratch and for different learning tasks. And they have also a collapse notebooks. Yeah, this 11 tasks you can try to follow. And also another nice tutorial is I think for these certain papers is you can try to read. I have some references in the notebooks. For example, you click here, so directly leads you to the paper. So that's initially semi-supervised classification with the graph convolutional neural network. You can get like more details here. For the code implementation, I suggest you to try to follow those collabs and the tutorials on YouTube, and I think you can really gain some knowledges in this touch geometry group. So also they have a Stanford tutorials, and I think there's another book is quite interesting. It's called a Graph Representation Learning. Yeah, this book by William Hamilton. So I will also put this link in my GitHub. You can try to download this book, and it has a quite nice introduction to starting from the graph machine learning, and then to the deep learning things. So those are the materials nice to follow, and I will update a README file in the GitHub. You can follow later after our session.
Okay, great. So hopefully the recording works as well, so that you can also replay it, and if you don't remember those references, you can view this video again as well. So this video should be on the ISCB website.
Yeah. And don't forget to start the project if you don't want to leave out the things. It has a collab and all the detailed implementation using the notebooks.
Yeah. So definitely, start from our GitHub and collab and go through it slower than we have today. Okay. And also our contacts are on there. If you have any further questions, you can reach out as well.
Okay. So our it's
on the GitHub.
Email,
on the collab.
Right here. Email us.
Yeah. Okay. Okay, great. So thank you, Ray and Evelyn, for delivering this tutorial. And thank you for those who are listening today. and asking very insightful questions. And yeah, good luck on your journey in tackling these machine learning problems and learning about GNNs. Okay, great. Thanks. Have a great day.